Good morning, students. Today we are having microbiology lecture number 12. Today we are going to talk about pathogens of spirochetosis and leptospirosis. If you remember, previous semester we studied morphology of bacteria, and in lecture number 1 we said that bacteria may be spherical shaped, rod shaped, and spiral shaped. So in this lecture, we will talk about those bacteria that are spiral shaped. They have a specific morphology in their shape. And the first pathogen that we are going to talk about is Treponema pallidum. This pathogen belongs to family Spirochaetaceae Genera Treponema, and species is Treponema pallidum. This bacteria causes syphilis. It is the causative agent of syphilis. Morphology of Treponema pallidum. They are slender spirals. You may look at this diagram. It is a smear. They are actively motile, very actively motile. They are non-capsulated and non-spore forming. This bacteria is gram-negative, but it is not stained by Gram's method because outer membrane is lipid rich. So we should use special staining technique for this bacterium. It is Romanovsky-Himze staining, and bacteria are stained in pink color. After Romanovsky-Himze staining. And we also may use silver impregnation, and because of it, bacteria are brown. They are brown. Their color is brown. If you remember, there is another bacteria that also has lipids in cell wall. It is Mycobacterium tuberculosis. So Mycobacterium tuberculosis and Treponema pallidum are not stained by Gram's method. This method is not used. The another one, cultivation or cultural properties. I want to pay your attention that Treponemas can't be cultivated in nutrient media. No one media is used, not simple media, not special media used. We can cultivate it just in the rabbit's testicles, not in all animals, not in any organ, just in the rabbit's testicles. Fermentative properties. I remind you that fermentative properties are used for identification of bacteria. What about fermentative properties of Treponema? Treponema aren't cultivated in media. That's why it is impossible to study fermentative properties. Actually, how do we study fermentative properties of bacteria? We cultivate bacteria in nutrient media with addition of some sugars, for example, some proteins. And if these sugars or proteins, if these substances are fermented, we can see that, for example, it ferments glucose, maltose, levulose, or sucrose. But In previous question, in cultural properties, I have just said that bacteria are not cultivated. That's why it is impossible to understand what do they ferment. Antigenic structure. Antigenic structure of Treponema includes three main antigens. The first one, it is lipid antigen of cell wall. This antigen is similar to cardiolipin, which is a phospholipid extracted from beef head. If you remember, in immunology, I told you about such condition as antigenic mimicry. Antigenic mimicry is similarity of antigens of different organisms, of human antigens and bacterial antigens, for example. So lipid antigen is similar. Lipid antigen of bacteria, Treponema pallidum, is similar to cardiolipin antigen of beef head, another organism. Protein antigen of cell wall is another antigen, and the polysaccharide antigen of cell wall is the third one. Virulence factors. This bacterium is gram-negative, so it produces endotoxin after disruption of bacterial cell. Then Treponema also have outer membrane proteins that are used by bacteria for adherence. The another one is Hyaluronidase. It enhances the invasiveness of bacteria. Invasiveness means ability to enter tissues, human tissues. Fibronectin that prevents phagocytosis. I remind you that virulence factors provide entry of bacteria in human body, its dissemination there, and destruction of human cells. They mediate all pathogenesis. So what about epidemiology and pathogenesis of syphilis? The only source of infection is sick men. Sick animals are not the source of infection. You may understand why. Why? Because of transmission way. Transmission way, in most cases, it is sexual contact during sexual transmission. Then natal transmission. I hope you remember, it's a transmission of infection from mother to fetus. And the last one is a contact, direct contact transmission. Firstly, I should say that it's very rare. It's rare in direct contact with vaginal discharge. Why is it so rare? Actually, Treponema pallidum are not resistant in environment. If Treponema pallidum present in environment with vaginal discharge or through with penis discharge, bacteria are dried with air during some minutes, during 10 minutes, after 10 minutes. So they die, these bacteria die during short period. So contact transmission is available, is possible if vaginal discharge with bacteria enters the mucosal membrane directly, mucosal membrane of vagina of another girl or woman, or maybe in coating in the skin. What about pathogenesis? How disease develops? What happens in human organism when bacteria enter there? Firstly, the first stage of pathogenesis is invasion of mucous membrane or skin. Bacteria invade by sexual transmission, mucous membrane of a vagina or penis or skin. Then the another stage is multiplication of bacteria at the site of infection. Bacteria multiply there. And the third one is formation of chancre. Chancre, it is the first clinical symptom that we can see in patient with a primary syphilis. Chancre is formed in the site of infection, in the site of bacterial penetration of the human organism. After primary syphilis, after formation of chancre, bacteria spread in the bloodstream and produce disseminated lesions, skin rashes. It is secondary syphilis. Let's talk about clinical manifestations of different stages. There are some stages of syphilis. These stages include primary syphilis, secondary syphilis, and tertiary syphilis or late stage. The first one is primary syphilis. I have just said this stage of disease is characterized by very specific clinical manifestation that is called hard chancre. Hard chancre develops at the site of inoculation of bacteria, the human organism, after 3-4 weeks. So, where can it occur? Hard chancre, look here. In this photo you can see hard chancre of the penis. Hard chancre may occur on the penis, in a vagina, or in tongue, in mouth, for example, in abnormal sexual contact. What is chancre? Hard chancre is a typically painless, it's not painful, it's painless, indurated ulcer with a clean base. This stage is highly infectious. Hard chancre consists of great amount of Treponema. In this stage we also can see regional lymphadenopathy. You remember lymphadenopathy, it's enlargement of lymph nodes. Regional means they are enlarged next to the site of infection. In these photos you also can see a hard chancre in penile region. Here it is painless ulcer with a clean base here or here. And in this photo you can see hard chancre in tongue. After 3 up to 6 weeks after primary syphilis, after chancre, secondary syphilis develops. Secondary syphilis develops because of dissemination or spreading of bacteria in the bloodstream, widespread of bacteria. The first and the main clinical symptom of secondary syphilis is a macular rash. It occurs on the skin, look here, and in this patient, or mucosal membranes. Mucocutaneous lesions also occur. Fever because of spreading of bacteria in the bloodstream and their partial destruction. Swollen lymph glands and sore throat. Sore throat is a specific symptom that is not characterized for secondary syphilis, but it occurs. If the patient doesn't take treatment, the last stage may develop. It is called tertiary syphilis or late stage. It develops within from 3 up to 10 years. It is characterized by formation of gummatose lesions. Gummatose lesions. What is gum? Gumma is a pathological lesion on the skin or in the mouth containing bacteria. Tertiary syphilis is also characterized by inflammation of the small blood vessels, that is called cardiovascular syphilis. Gummatose lesions or gumma are formed in blood vessels. And neurosyphilis, it is a final stage or final phase of tertiary syphilis, final result, that is characterized by inflammation of cranial nerves and deafness and full destruction of nervous system. We have just said about primary syphilis, secondary syphilis and tertiary syphilis that develop because of sexual transmission of Treponema pallidum. And you remember that there is another transmission way. It is natal transmission or transmission from mother to fetus. There are two clinical forms of congenital syphilis. The first one is called early congenital syphilis, and the second one is called late congenital syphilis. Early congenital syphilis is characterized by symptoms that occur during 2 years after birth. What are the symptoms? These are symptoms of primary and secondary syphilis. These are mostly lesions, macular rashes or lesions in mucous membranes or on the skin. Late congenital syphilis is characterized by clinical symptoms that appear during 15 years after the birth. Congenital syphilis occurs when a child is born to a mother with primary or secondary syphilis. Actually, if mother has tertiary syphilis, bacteria are deposited in gumma or in granulomas in internal organs. So such transmission is impossible. Treponema pallidum may cross placenta, so they cause infection. When does early congenital syphilis develop and when does late congenital syphilis develop? It is not known. They say that clinical form, early or late, depends on stage of infection of mother. Stage of infection is primary or secondary, and which organs are infected, and amount of bacteria in mother's organism. So we have just said about pathogenesis. Now, how to diagnose syphilis? Firstly, for diagnosis we should collect specimen. What is specimen? The choice of specimen depends on the stage of infection, depends on clinical form, stage of infection. In primary syphilis, we should collect exudates. Exudates are excretions from hard chancre, fluid from surface lesions is collected in secondary syphilis. Blood may be collected just in secondary syphilis. In primary syphilis, we must not collect blood. Bacteria are absent there. We will not be able to find Treponema there. Secondary syphilis is a disseminated infection, so we should collect blood. Serum is collected for serological tests, but you should remember that antibodies are absent in serum of the patient during incubation period, during first 3 weeks. And cerebrospinal fluid is collected in tertiary syphilis. Which methods are used? The first one, it is presumptive, that is used for presumptive diagnosis, is microscopic method. Microscopy has great importance in diagnosis of syphilis, but it is not light microscopy. It is special microscopic technique. It's dark field microscopy, when we can see bright bacterial cell on the dark field, black background. And another one is direct fluorescence antibody microscopy. We use fluorescence labeled antibodies, or antibodies that are labeled with fluorescence dyes or fluorescence paintings. They are bound to bacteria. They cover all the surface of Treponema, and we can see brightening of Treponema in the background. Actually, Treponema has a very specific morphology, very specific shape. They are slender spiral-shaped bacteria. They are isolated from penis or vagina, so other bacteria that have such form, such shape, are not able to live in vagina or penis. So microscopy allows to make presumptive diagnosis. But sure, we should use other methods. The another important method is serological method, or serological tests. Serological tests include two type of tests. The first one is non-treponemal tests. For these tests we use cardiolipin antigen of beef head. When I say to you about antigenic structure, I said that lipid antigen of Treponema pallidum is similar to cardiolipin antigen of beef head, so we use it. What are these non-treponemal tests? This is complement fixation test, or it is also called Wassermann test, because of name of doctor who suggested to use it. And another one is VDRL test, or microprecipitation test. We will talk about mechanism of complement fixation test and precipitation test in immunology. VDRL test is called VDRL because it was developed in Venereal Disease Research Laboratory in the New York and USA. It's also non-treponemal test that use cardiolipin antigen. Treponemal tests is another group of tests. They use antigens of living bacteria, antigens of Treponema. And these tests include ELISA enzyme-linked immunosorbent assay and Treponema pallidum hemagglutination assay. When should we use non-treponemal tests? When should we use treponemal tests? It's very easy to understand. Non-treponemal tests are used for screening or diagnosis of large number of sera. When there are a lot of patients for diagnosis, all sera that are collected from patients firstly diagnosed by with using of non-treponemal tests, because it is cheaper, it's not so expensive. And treponemal tests are used to confirm diagnosis of the positive non-treponema test. If the first test in patient is positive, diagnosis must be confirmed by treponemal tests. Bacteriological method is not used, and you understand why, because bacteria, Treponema pallidum, can't grow on nutrient media. And PCR, sure, it's also important method for diagnosis of syphilis. What about prevention, prophylaxis, and treatment? Vaccines are not available for prophylaxis of syphilis. There is no one vaccine in the world. There is just general transmission. It's protective sexual contacts by using of condoms that help prevent transmission. You shouldn't be shy here. You shouldn't be shy. When you talk about it, when you will be doctors, you should inform your patients, especially those patients who don't like to use condoms. Treatment. The first antibiotic that was used, and it is used these days, it's penicillin G, benzathine penicillin G. It's effective in treatment of syphilis. If a patient has allergy to penicillin, we may use doxycycline or tetracycline. If we make conclusion about syphilis, we can say that this disease was one of the most famous because people never had condoms early years, and in ancient times, for example, earlier syphilis was spread in cities and towns that are port cities that are located along the seas or oceans. When ships with soldiers, when ships with sailors come back to the port in some city, and a lot of women were waiting there for these sailors and soldiers. So another pathogen is Borrelia. Biological properties of Borrelia. This pathogen is also belongs to family Spirochactaceae. It has the same shape. Genus is Borrelia, and there are two species that are clinically important. The first one is Borrelia recurrentis, that causes epidemic and endemic relapsing fever, and Borrelia burgdorferi, that causes Lyme disease. Morphology. Borrelia. Borrelia are called Borrelia because this bacteria was discovered by Borrell. These are large motile spiral bacteria with from 3 up to 10 loose coils. You can see in this photo shape of bacteria, morphology of bacteria. These are macrophage and these are Borrelia. They are also gram-negative, non-capsulated, non-spore forming, and actively motile. They are spiral, so it's very easy to move because of such shape. Bacteria also poorly and not stained. By gram staining, we use special staining technique. It's Romanovsky-Himze stain. And bacteria, Borrelia, is stained in blue-violet color. Unlike Treponema, that is stained in pink color. Cultural properties. This bacteria is microaerophilic. Microaerophilic. If you remember, microaerophilic needs less amount of oxygen. Less amount of oxygen. Cultivation of Borrelia is very difficult. Nutrient media are not used. No one special or simple medium. This bacteria can grow on chorion-allantoic membrane of chick embryos. You can see in this picture that bacteria are inoculated from via syringe into chorion-allantoic membrane of chick embryos. They develop there. And we also can inoculate this bacteria into animals, in mice or rats, intraperitoneally, in abdomen. Fermentative properties. They are not active in fermentation, because they are not cultivated in nutrient media. We can't study it. Antigenic properties. There is one important property of Borrelia. It is antigenic variation. Antigenic variation is a unique property, therefore occurrence of relapses in the disease. Bacteria changes its antigen several times. What about virulence factors? This bacteria is gram-negative, so it produces endotoxin. Borrelia has adhesins. These are proteins that are effective for adherence of bacteria to cells. Invasiveness is another one. Invasiveness is the ability to enter the human tissues. Invasiveness occurs because of motility of bacteria. They are very motile, so it provides invasiveness and surface membrane proteins. And antigenic reaction that I have just said is also virulence factor, and it leads to escape from immune clearance, from immune response, because immune system is not able to produce memory cells and lifelong immunity. Not able to cause lifelong immunity. What about epidemiology and pathogenesis of relapsing fever? There are two diseases that may be caused by Borrelia recurrentis. It's epidemic relapsing fever, and another name is louse-borne. You may understand that the main vector is louse. Endemic relapsing fever is also called tick-borne, because the main vector, the only vector here, is tick, ixoid tick. Source of infection. In epidemic relapsing fever, this is sick man, because louse may live just on the man. It's not fleas. Don't mix, please, here. In endemic relapsing fever, sources are sick animals, mostly wild animals, rodents, small mammals, mostly wild animals. These are not house rats, not house rodents, not sewer rats. Transmission, it's biting of lice in an epidemic relapsing fever and biting of ticks in endemic relapsing fever. What about pathogenesis? Incubation period lasts from 2 up to 10 days. Bacteria enter the blood through biting of lice or biting of ticks. Immune response is activated and macrophages and antibodies destroy Borrelia. You remember that Borrelia is gram-negative bacteria that releases endotoxin after bacterial cell disruption. In the presence of large amount of endotoxin, our patient has high fever up to 40 degrees Celsius. This is so-called febrile period that lasts several days. Bacteria, Borrelia, are deposited in liver, change their antigenic structure. I have said about antigenic variation. Change their antigenic structure. So, immune system recognizes this pathogen as another pathogen because previous antibodies are not effective. Antigenic structure is changed. And immune system provide production of new antibodies. This period is afebrile. There is no body temperature, there is no fever. It lasts about 10 days. During this period, when antibodies are produced, when bacteria change their antigenic structure, when antibodies are produced. After production of antibodies, you know that why is it 10 days? You know that maximal amount of antibodies is on the 7th day after starting of disease. I've said about it in immunology. You studied this information. So it lasts about 10 days. New antibodies destroy Borrelia, and once more it causes release of endotoxin, and once more causes fever and febrile period. Then bacteria are deposited in liver, change their antigenic structure, cause production of new antibodies and a febrile period. It repeats once more. And such relapses, such processes repeat about from 3 up to 10 times. This is also characterized by hepatosplenomegaly because of deposition of bacteria in liver and neurological complications. Another disease that is caused by Borrelia is Lyme's disease. This disease was identified in 1975 in Lyme, Connecticut, in the United States of America. It is caused by another species. Some species are able to cause Lyme disease. Borrelia burgdorferi is the most famous, is the most spread. Borrelia garini and Borrelia afzeli. What about epidemiology of Lyme disease? Sources of Lyme disease are both wild animals or domestic animals. It may be deer, rodents, dogs, sheep, birds, cattle. Any animal that can be bitten by exotic tick, because the vector is exotic tick. It's transmitted by biting of ticks. Infection occurs by discharge of the gut content during biting. Actually, exotic ticks are only vectors. These insects are not sick. They don't have clinical symptoms of Lyme's disease or relapsing fever. They are vectors. Borrelia is a normal microflora of exotic tick intestine. So before biting, ticks are ticks release feces from the gut, discharge from the gut, and release bacteria with their feces. Then after biting, uh, the patient starts to scratch site of biting and inject this bacteria into the biting site, into the blood. Pathogenesis of Lyme disease. There are three stages of Lyme disease. The first one is localized infection. Localized infection is also called erythema chronicum migrans. It is a macule, red spot at the site of bite with redness and induration. The second one is disseminated infection. Disseminated infection is when bacteria spread in the bloodstream and it causes fever because of destruction of bacteria, headache, myalgia, arthralgia, lymphadenopathy, and skin lesions. Meningitis and arthritis also complications of disseminated Lyme disease. And the last one is a persistent infection that is characterized by chronic skin lesions, chronic neurological symptoms, and chronic arthritis. In this photo, in this diagram, you can see erythema chronicum migrans. It is papule that is formed, that is manifested at the site of biting, of ixotic tick. It is called erythema chronicum migrans because it can move on the skin from one side to another side when bacteria multiply under the skin. What about diagnosis, treatment, and prophylaxis of relapsing fever and Lyme disease? The only specimen that may be collected here is blood during fever. Why can't we collect blood after fever? Because bacteria are absent in the bloodstream. You remember they are deposited in liver and change their antigen structure. So blood before or during fever. Before fever, the patient doesn't know about his disease. Which methods are used for diagnosis? The first one is a microscopic method. It is dark-field microscopy. Like here we can see a bacteria on dark, on almost black background, and Romanovsky-Himze staining. You can see that here bacteria are stained in blue, violet, in violet color. Gram staining is not used, you remember. The second one, biological method. It's inoculation of blood into white mice and then preparation of smear. So we collect blood of the patient, inoculate into mice. Mice are infected, develops relapsing fever. We collect blood from this animal and observe bacteria there. It means the presence of Borrelia in specimen. PCR is also used for diagnosis. Serological and bacteriological methods are not available. Why? You remember, for serological tests we should collect serum for detection of antibodies, but Borrelia have antigenic variable structure. It is impossible to know which antigen will be in this patient. Bacteriological is not used because bacteria are not cultivated. What about treatment and prophylaxis of relapsing fever and Lyme disease? There are no any vaccines. Vaccines are also not available because of variable antigens. And the total or general prophylaxis includes control of vectors, control of spreading of lice or ticks where people live. Treatment includes broad spectrum antibiotics These are tetracycline, chloramphenicol, penicillin, and erythromycin. And the last pathogen here is Leptospira. Family Leptospiraceae, genus Leptospira, and this bacteria causes leptospirosis. Leptospirosis. This is also spiral bacteria with closely coils and hooked ends. They are gram-negative but poorly stained, very actively motile, non-spore forming, non-capsulated. There is a special staining techniques because gram staining is not used. Special staining techniques is silver impregnation technique, and bacteria are stained in brown color. And we also may use dark field microscopy like here, or fluorescence microscopy. When smear is treated with fluorescence antibodies that bind to bacteria, and we can see brightening of bacteria on black background. What about cultivation? This bacteria is aerobic or microaerophilic. Need a decreased amount of oxygen, and this bacteria is the only that can be cultivated on media. Treponema and Borrelia are not cultivated. Leptospira may be cultivated in Fletcher's media. Fletcher's media consists of beef extract and rabbit serum. We also can cultivate Leptospira in chick embryo. So growth of bacteria is longer, and it's observed after 1-2 weeks. Bacteria aren't active in fermentation. We know just about such fermentative properties, that is oxidase test is positive and catalase test is positive. Antigenic structure. Outer envelope of bacteria contains large amount of lipopolysaccharides, so it is the first antigen and the only antigen of Leptospira. Antigenic structure of Leptospira varies from one strain to another, so there are about 200 variants of antigenic structure of Leptospira, and that's why it forms the basis of serological classification. 200 serological variants that are distinguished in structure of lipopolysaccharide. What about virulence factors? This bacterium is gram-negative, so it produces endotoxin that damages epithelial cells and disrupts the capillaries and causes bleeding. So the first, one of the most widespread manifestation is bleeding, internal bleeding, for example. The another one is fibrinolysin. It dissolves clots of fibrin, so Leptospira penetrates into the bloodstream. You know that fibrin is a protein of blood plasma that block bleedings. So bacteria destroy fibrin and bacteria may penetrate the bloodstream. Hemolysin, you know, it destroys red blood cells, erythrocytes, causes multiple vascular disorders. And the last one is plasma coagulase. It causes coagulation of plasma proteins, multiple clots develop, leads to ischemia. Ischemia is poor blood supply, and then it leads to hypoxia, absence of oxygen, leads to ischemia of organs and system. Epidemiology and pathogenesis of leptospirosis. Source of infections are animals, wild animals mostly. Rodents. They discharge bacteria with urine during urination. Transmission. It is leptospirosis transmitted by direct contact with water contaminated by animals' urine through cuts or skin abrasion. So, when we swim in the river or in the lake, where can be present, where Leptospira can be present, so potentially we may be infected. Waterborne through swallowing of water with bacteria, or foodborne through ingestion of contaminated water or food. Incubation period of leptospirosis lasts from 1 up to 2 weeks. Leptospira are present in the water with animal urine, I've said about it. Bacteria enters through breaks in the skin, cuts and abrasions, and through mucous membranes. Then enters also may enter through mouth, nose, conjunctiva. Then bacteria enter the bloodstream and multiply in the bloodstream and produce fever. Bacteria enter into the liver, kidney, spleen, and meninges in internal parenchymatous organs. There, bacteria secrete virulence factors. We know what are they: fibrinolysin, endotoxin, hemolysin, and plasma coagulase. These virulence factors, let's come back. And there, bacteria produce hemorrhages because of endotoxin, necrosis because of plasma coagulase, necrosis in tissues, and initiates dysfunction of these organs, disorders. One of the most severe clinical manifestation of leptospirosis is called Weil's syndrome. Weil's syndrome is a severe form of leptospirosis that causes continuous fever, long fever, kidney damage, so we can see blood in the urine and painful urination, and liver injury. In these diagrams you can see the patient with Weil's syndrome. Dark urine is observed, rashes because of bleedings, and yellowing of eye globe. Why? Also because of bleedings. Microbiological diagnosis, treatment, and prophylaxis of leptospirosis. Once more, yellowing of eye globe also, and yellow epidermis occurs because of liver destruction. Specimen includes collection of blood, urine, and serum, and cerebrospinal fluid or tissues. So those material, those fluids that may contain Leptospira. Which methods are used for diagnosis? The first one is microscopic. It's dark ground microscopy of the blood specimen in first 8 days. Bacteriological methods include inoculation of bacteria on Fletcher's medium, then serological tests that includes complement fixation test, agglutination test, and ELISA, enzyme-linked immunosorbent assay, and polymerase chain reaction, PCR, is also used. What about treatment and prophylaxis of leptospirosis? All patients, those patients who are infected with leptospirosis produce serovirus-specific immunity after disease. What does it mean? Lifelong resistance, lifelong immunity to serovar, serological variant. I have said that there are about 200 serological variants. So patient may have immunity to one serological variant but may be infected by another serological variant in another city, for example. For prevention, we may use killed vaccine. It's used for agricultural workers that contacts with water with that potentially may be infected with animal urine. For treatment, we also should use broad-spectrum antibiotics, for example, doxycycline, ampicillin, and amoxicillin. So that's all. In this lecture, there are just 3 pathogens. This lecture is easier than previous ones. And thanks for your attention.